Hello. Today I will discuss about the microscopy. Microscopy is the branch of science which deals with study of microscope. Now, what is the literal meaning of microscope? What is the definition of microscope? Microscope, it is an instrument consisting a lens or a combination of lenses used to observe or to magnify the object which is under observation. Now, why we require a microscope? Microscopes are required to observe very minute things. Means what? Those things which cannot be seen by naked eyes. Generally, microscope, they are of different types, but basically we can classify the microscopes into two categories. One, that is a light microscope, and second, that is electron microscope. Light microscopes, they are further divided into two types. One, bright field microscope and dark field microscope. Now, what is meant by light microscope? So, light microscope, it is the microscope which requires light as a source of illumination, and the electron microscope is the microscope which requires electron or a beam of electron as a source of illumination. Light microscope is further divided into two types: bright field microscope and dark field microscope. Bright field microscope are those microscopes in which we get a field brightly illuminated against the dark object. And second thing, that is a dark field microscope. In the dark field microscope, we get the object which is brightly illuminated against dark background. Now, when we try to study the microscope, we should be familiar with the two basic concepts or the properties of two things. One, that is light. We should know the properties of light like frequency, amplitude and wavelength. And second, that is properties of optics or lenses. These two things are very important to understand the basic concepts of microscopy. Simple microscope is another type of microscope in which they are made up of a single lens. So further we can classify in different manner that microscopes are of two types: the simple microscope and compound microscope. Simple microscopes, these are the microscopes which are made up of a single lens, which are generally used for dissection purpose. And second, that is a compound microscope. The word compound means that it is made up of more than two lenses. So any microscope which is made up of two lenses, or which consists two lenses, a combination of two lenses, that is known as a compound microscope. Now, the basic difference between a light microscope and the electron microscope is light microscope, it has low magnification power as well as low resolving power. But in case of electron microscope, it has a high resolving power as well as high magnification powers. And in terms of cost, they are also highly costly, or they are very costly. That means ordinary lab or ordinary people, they cannot purchase this microscope. So generally the resolving power is another concept. Resolving power is what? It is the ability of the lens to distinguish two closely placed objects as a separate one. But resolving power, it is dependent on various things. that is what the wavelength of light and the refractive index. Wavelength of light, as the wavelength of light becomes narrower or smaller, the resolving power of the microscope increases, and as it goes on increasing, the resolving power of microscope decreases. This is the reason why electron microscope, it has a more resolving power than the ordinary light microscope. And second thing is what? The refractive index. So numerical aperture. Generally, when we talk about the compound microscope, this compound microscope uses three types of lenses. These lenses, objective lenses, they are known as objective lenses. These objective lenses are low power objective, having a magnification power of 10x. High power objective, that is a magnification power of 45x. And the micro objective lens, which is generally used for observation of very small things like bacteria, that is called as oil immersion lens. Okay. Now two lenses, out of these three lenses, two lenses like a low power objective and a high power objective, they are operated in air. That's why their numerical aperture value is less. Generally numerical aperture value, it is engraved on the barrel of objective. When we use low power objective, the numerical aperture value is near about 0.25. When we use high power objective, the numerical aperture value, it is around 0.65. It cannot go beyond 1, okay, because these two lenses are operated in open air. But when we use oil immersion lens, so oil immersion lens is that lens which is used by many microbiologists. It has a magnification power greater than these two lenses, that is low power and high power. Its numerical aperture value is also higher, it is around 1.25. It has maximum numerical aperture value, that is 1.25 as compared to the other two lenses, because it is not operated in open air. Okay. While using these lenses, we have to immerse the lens in the specific oil which has the refractive index value equal to the glass, near about 1.52. Okay. So when we try to observe any object under the microscope by using oil immersion lens, then we should put a drop of oil on the object which is placed on the slide, and then we have to immerse the oil immersion lens in that drop and try to focus the objects. So as there is no gap between the object and the oil immersion lens, that's why the diffraction of light rays becomes very small, and that's why we get a maximum magnification power or magnified image. Now when we use electron microscope, the electron microscopes are broadly classified into two common types. One that is called as a TEM. TEM means transmission electron microscope, and second one is SEM, scanning electron microscope. Now literal meaning of these two types of microscope is what? When we say TEM, transmission electron microscope, means in this type of microscope we get the image of the object obtained from the transmitted electrons. In case of scanning, only the surface structure of the object, it is scanned by the electron, and from the secondary electron the image of the object is obtained, but that is very fine image. So in case of transmission electron microscope, when we get the image, that image consists all the intracellular details, but when we try to observe the image of scanning electron microscope, we can see only the surface details, minute surface details when we try to observe the object through the scanning electron microscope. Now the basic difference between a light microscope and electron microscope. Light microscopes, they can operate in open air, but when we try to use the electron microscope, we have to use it in a very sophisticated conditions. They require a separate room, separate power supply, as well as vacuum conditions should be there. In case of electron microscopes, vacuum in the column, it is maintained because air consists so many dust particles than gas molecules. So if these gas molecules or dust particles, they remain in the path of electron beam in case of electron microscope, then there may be the generation of heat due to the collision of electrons with those dust particles or gas molecules, and that can cause the deterioration of the object. This is the reason why vacuum is created in the electron microscope. Second thing, we cannot, whatever the lenses those are used in electron microscope, those lenses are electromagnetic lenses. Means these are the magnetic lenses surrounded by the coils of wires which are provided with the currents, and by changing the currents we can magnify the object, we can focus the object. We cannot use the glass lenses in case of microscope, electron microscope, because electrons, they cannot penetrate the glass, and this is the reason why we cannot use the glass. And another thing which is different from the light microscope is when we try to mount the object, that object should be mounted on the specific grid. That grid is called as a copper grid. It is small metal disc which is made up of copper, which is highly perforated, and on that the sample is loaded, and we try to observe that sample which is loaded on the copper grid. Another thing, we cannot directly view the image with the electrons like the light microscope. When the light enters in our eye, that form the image of the object on our retina. But in case of electron microscope, those images, they are first of all formed on the fluorescent screen or X-ray screen, and that image we can observe through the viewing window. Now this is about the basic concepts of microscopy. Thank you.